Hello, I'm Martin Blaser. I'm the director of the Center for Advanced Biotechnology and Medicine, or CABM. Today, I have the pleasure of speaking with CABM resident faculty member, Dr. Jennifer Mulle. She's an associate professor in the departments of psychiatry and neuroscience and cell biology at Robert Wood Johnson Medical School. So Jen, nice to talk to you. Can you tell us a little about your education and the early parts of your career?
Yes, I would love to. Thank you for having me. I'm thrilled to be here. So I grew up, as you know, on Long Island in New York. When I graduated high school, I went to Johns Hopkins University. I was pre-med at Johns Hopkins for all of 5 minutes. And pretty quickly, I realized that pre-med wasn't quite right for me. And I spent some time sort of trying to figure out what I wanted. I didn't know by the time I graduated. And so I worked for 5 years. I worked in various lab environments. I was in an academic lab, a government lab, and then ultimately a drug company. And it was through those experiences that I got exposed to genetics and that I realized that genetics really was where I wanted to be. And so after 5 years, after taking a break for 5 years, I did a master's degree in genetic epidemiology at Johns Hopkins. And then I did a PhD in human genetics proper. And then I did a postdoctoral fellowship at Emory University. I took a faculty position at Emory, and now I'm at Rutgers.
Great. So how long have you been here?
I got here in September, so I'm going on 5 months now.
So you're an infant.
A little bit, yeah.
Right. So can you tell us what's the main focus of your lab? What are some of the key questions you're exploring?
Sure. So part of the reason why I took 5 years off in between college and grad school was because schizophrenia actually runs in my family. My dad had a sister who had schizophrenia. There are a number of other relatives. And so I understood at a young age that I was at risk. And I wanted to take some time. I wasn't really sure. I mean, a lot of my sort of high school years and time in my 20s was spent wondering, am I going to get sick? What's going to happen to me? And so it turns out that risk for schizophrenia peaks around the age of 20 to 25 and then falls off precipitously. And by the time you reach the age of 30, if you haven't gotten schizophrenia, you're unlikely to get it. So I got to be about 28 and I hadn't had any hallucinations or delusions. My relationship to reality was still somewhat intact. And so that's when I decided to go back to school, but I was really motivated by this question of genetic risk for schizophrenia. And so for my master's degree, I did linkage analysis, looking at family-based patterns of schizophrenia. For my PhD, I did a sort of association study looking at linkage peaks on chromosome 13, asking whether there was any relationship between genetic variation and schizophrenia. Both of those studies were spectacularly unsuccessful. We found nothing. Then I moved to Emory and started looking at DNA dosage changes, whether those are associated with risk for schizophrenia. And we found a deletion on the long arm of chromosome 3. It's 1.6 megabases. It contains 21 genes. And there's something about missing that segment of DNA that renders you extremely susceptible to schizophrenia. People who have that deletion are 40 times more likely to get schizophrenia than someone in the general population. And with that discovery, the rest of my career was set. I knew that I wanted to understand what that deletion does and why people with that deletion are so susceptible to schizophrenia.
Wow, that's a great story. I never heard you say it that way. And so it starts from you and your family, and then you find this tremendously strong association. And that, by the way, that's the key for everything in genetics. You have a strong phenotype like schizophrenia. You can really understand things. So
yeah.
Great. So that's really the question. So how are you approaching it now?
Yeah, so it turns out it's really hard to do. So there's this deletion, and I want to understand what this deletion is doing. And so the first thing I need human humans with the deletion. And so I started a registry to collect individuals with the deletion. And in the registry, we now have about 240 individuals with the deletion. And with the registry and with these human study subjects, we've been able to describe what the phenotype is. And it turns out that individuals with the deletion don't just have schizophrenia. They have some cognitive disability, they have some social disability and risk for autism, they have attentional problems, executive function deficits, and risk for ADHD. And then, of course, later in life, they are at risk for schizophrenia. And we can even see with our various phenotyping tools, we can see those phenotypes, those kind of early phenotypes of schizophrenia emerge long before the onset of the disorder. And so that's one way that we that we explore the sort of phenotype, or that we're asking these questions. We also have cellular models from our patients, and we do molecular and cellular based assays to ask what the deletion is doing. And then for more complex studies, in vivo studies, we have a mouse model of the deletion, and all of those resources are now at Rutgers, which is really exciting.
Congratulations getting through the paperwork.
Yeah, I think there's still some to do, but
and one of the great things about Zoom is that I now know that you have a dog.
We actually have 3 dogs.
Three dog nights. So,
yeah.
let me ask you a question. The 240 people, how were they identified that they had the deletion?
That's a great question. So what happens is, I mean, do you have kids?
Yes. Mm-hmm.
Yeah. So you know this, right?
And
so
my kids are about your age. So.
Awesome. So my kids are a little younger. I have kids who are 10 and 12. And when you have kids, you know, right? One of the first things you ask is like, well, when are they going to sit up? And when are they going to walk? And when are they going to talk? Right. And so kids with 3q29 deletion syndrome do all of those things. They just do them on a very delayed schedule. And so a baby may not sit up until 8 months, and they may not walk until 18 months or 2 years. And a lot of these kids have speech delay, and they have some other things. And parents go to the doctor and say to their pediatrician and say, my baby isn't talking yet. And the pediatrician says, maybe it's time to do a genetic test. And when that genetic test goes in, about 1 in 1,000 times when a child is referred for genetic testing, it comes back with 3q29 deletion syndrome. So parents get results from a genetic test that say your baby has 3q29 deletion syndrome. And you'd think that would be amazing. It would be an answer, right? Like I know, but it turns out that that's almost, for a while before we got involved, there was very little information. And so parents would go to the clinic and they would say, well, I have this result, I have this answer, my child has 3q29 deletion syndrome. And the clinic would say, wow, this is the first case we've ever seen, or we've never seen one of these before. We don't, you know, and so it really came with very little information. And so through our work, we feel very grateful that we've been able to provide some answers to families. We can say now, these are the things that your child is at risk for. These are the things that are helpful. And while we don't have answers yet about treatment, we're hopeful that we're going to get there.
Sure. So if it's 1 in 1,000 in kids who are getting genetic screening, that's not your random population. If you looked at the random population, would it be 1 in 40,000?
Yeah, exactly. Good. It's about 1 in 30 to 1 in 40,000. So quite rare.
Mm-hmm. Got it. I see. Can you detect it through 23andMe, for example?
So we've been trying for a while to get access to 23andMe data. We would love to know what the frequency is in 23andMe. We know in other large samples, like for example, the UK Biobank is a large sample of several 100,000 individuals, but it seems to be depleted for individuals who have rare copy number variants. And we suspect that that's because the amount of wherewithal that it takes to join a research study, to consent, to go in, to do all of the things, that's hard for people who have 3q29 deletion or 22q deletion or any of the other rare genetic disorders. And so you have to be somewhat intentional to recruit those people.
Yeah, great. So interesting. Thank you. So what new project are you working on now? What's particularly exciting to you?
So what's really exciting to me, Marty, is, so when I say cerebellum, what's the first thing that you think?
Movement.
You think motor disorders, right? You think ataxia, you think, tremors, stuff like that. So it turns out that the cerebellum is probably so much more than that. That just like it exerts control over motor function, the cerebellum also probably exerts control over things like executive function, sort of social functioning. But we sort of are still struggling to understand that. And the cerebellum, frankly, has been a bit of an understudied piece of the brain. So we were lucky enough to bring in individuals with 3q29 deletion syndrome and do some deep phenotyping to ask, what do they look like? And in the process of this, we also did some neuroimaging studies. And we found that their cerebella are remarkably sort of impacted. They have individuals with 3q29 deletion syndrome have a much smaller cerebellum than you would expect. And you don't have to be a neuroimaging specialist to see it. If you or I were looking at a scan, we could see how small that cerebellum is. And it turns out that 3q29 deletion syndrome is not the only syndrome that's associated with neurodevelopmental psychiatric disorders that also seems to have cerebellar pathology. There are now a whole host of disorders that look like they also are related to the cerebellum. And so the cerebellum is emerging as this sort of central unit or key kind of structural organ that may be related strongly to risk for autism, schizophrenia, intellectual disability, executive function disorders. And so that's what I'm really excited about, is that we could potentially add to that literature. And if it turns out that 3q29 deletion syndrome really sort of, if the primary impact is to the cerebellum, we now have a place to start looking for the molecular and cellular pathology.
Great, great. So when are we going to give antibiotics to the 3q29?
We're totally going to do that. We're going to give them antibiotics, and we're going to see if their behavioral phenotypes get worse. I love that.
Great. So now tell me, which school are you in? Which departments are you in?
So I'm in the medical school, in Robert Wood Johnson Medical School. I think my primary appointment is in the Department of Psychiatry. And I have a secondary appointment in neuroscience and cell biology. I'm also part of the Brain Health Institute, and I'm a member of the Center for Psychiatric Genetics and Addictions. So, and I'm already, we mentioned I haven't been here very long, but I already have multiple collaborations springing up. It's been such a wonderful experience to connect with other researchers. It's been great.
Great, terrific. And so that's other researchers, but can you tell us how you interact with people in your lab, students, postdocs, other trainees?
Yeah, so I think having trainees is like one of the joys of my life. Being able to cultivate and develop new scientists feels like such a privilege. So I focus a lot on goals. To me, how long someone is in lab and when they're in lab and what hours they keep is really up to them. It's up to them to manage their time. We have things we got to get done, and you get them done however you get them done. You work in the way that's best for you, but we really want to see progress. We want to see performance. I also strongly encourage my trainees that they have to live their whole life, right? Science is important, but everything is important. Your relationship with your significant other, your relationship with your family, your hobbies, self-care, all of that's important. And I feel firmly that the time that you spend in lab is really important, but the time that you spend outside of lab is just as important. And when you spend good time outside of lab, you come back refreshed and energized. And I think, you're productive. So that's how I tend to interact with that's my sort of lab philosophy. And it should be fun, right? Like
Right.
We do such great things. It should be fun.
I'm signing up for your lab.
I would love to have you. You're
That's
totally welcome anytime.
really great. So I guess my last question is, do you have other roles at Rutgers or in the scientific community that you want to comment about?
Yeah, so I'm lucky to be part of a collaborative effort that's funded by NIMH. It's called the Genes to Mental Health Network, and it unites a whole bunch of researchers, really from the US, but also outside the US, who are all studying rare genetic syndromes. And having that sort of collaborative way to come together and kind of unify or harmonize the ways that we're all approaching rare genetic syndromes has been really exciting to me. And so that's one of the things that I'm most excited about outside of Rutgers that I belong to.
Great, great. Well, Jen, thanks so much for sharing a little about your life and your art, your art of science with everyone. I really wish you well as you progress through the years at Rutgers.
Thank you so much, Marty. It's
Thank you.
delightful to be here.
Thank you.